So, Federico Licastro from the University of Bologna in Italy. Professor Licastro is professor of immunology and medical doctor. He's interested in Alzheimer disease and Down syndrome research and working on immune system. Please, Dr. Licastro.
Thank you for your kind introduction. I'm very pleased to be here and be part of this group. I hope we'll be successful in proposing something very new in the Down syndrome and also Alzheimer's disease field. My expertise is basically in immunology and immune genetics, and so I will talk about what we know about immunology and immunopathology of Down syndrome subjects. There are several studies performed during the last 20 years showing that there are several immune defects well observable both in children and adults with Down syndrome. And I summarized the most important results, which have also a clinical and biological consequences. I just choose to divide three small parts about the T-cell compartment, which is a very important part of the adaptive immunity or antigen-specific immunity. So we have subjects with Down syndrome usually show an accelerated thymus involution. It starts early, and the thymus is start to be reduced in size, also produce less thymic hormones, and the end results of this defective biological development of the thymus is a reduced T-cell repertoire, which is very important because our repertoire is very important because orchestrate our defenses against bacteria, virus infection, protozoa, and so on. So if subjects with Down syndrome have a reduced T-cell repertoire, they will be more exposed to infection. In fact, that is the reality. In the peripheral blood, we can see several alterations of immune responses. For example, there is a decreased number of CD4 T cell. This population are regulatory T cell. They help the immune responses. They regulate B cell function. They regulate macrophage responsiveness, and so on. There is, since the repertoire is defective, we can find in the peripheral blood a decreasing number of naive T cells, which has this particular phenotype. Naive cells are very important because they respond to new insults. They respond to new infection. If the number of naive T cells is decreased, the Down syndrome subjects are more exposed to infection. On the other hand, there is an increased percentage and number of memory T cells, both with CD4 helper and CT8 cytotoxic phenotype. We found, we published 3 or 4 years ago, there is also, I saw a recent paper confirming the data, that there are there is an increasing number of pre-apoptotic T cells in the peripheral blood. They express this particular marker annexin 5. So if the number of pre-apoptotic cells is increased, you have a decreased T cell proliferation, decreased T cell response to antigen challenge, so you have a defective clone expansion. If we look at the B cell compartment, cells that produce antibodies, which we have in the blood and in other biological fluid, We found we can see a decreasing number of B-cell differentiation, a decreasing number of maturation in the bone marrow, and you can find also a decreasing number of mature B-cell in the peripheral blood, and several alterations in the immunoglobulin levels in the plasma and serum of children and adults with Down syndrome. Also, the natural immunity is not quite normal. If you compare the natural immunity in Down syndrome subjects and in controls comparable with age, of course, you can find some typical alteration. For example, there is an increasing number of pro-inflammatory monocytes expressing a particular phenotype like CD4 and CD16 positive. And that means, this result means that monocytes are primed to produce more inflammatory factors like cytokines, for example. There is also, you can find an increasing number of natural killer cells. Those are, these kind of cells are very important because they respond against mutated cells, so they control tumors, for example, or they kill virus-infected cells. So the number is increased, but the function is defective. So they are not very efficient in doing their job. There are also altered cytokine levels easily detectable in the plasma level, in the plasma and the blood. For example, there is increased level of IL-6, its soluble receptor, increased concentration of IL-7, interleukin 15, and also factors controlling chemotaxic of the transfer of lymphocytes and leukocytes from the blood to the tissue, or the activation of endothelial cell like VEGF or MCP1. Another several chemokines controlling the activation of endothelial cell are also abnormal in the plasma level of children and adults with Down syndrome. So what kind of consequence we can expect since we found an impaired immune response in Down syndrome? That is very well known. The consequence of this is that acute and tendencies to be exposed to acute and chronic infection, especially infection by virus. This picture was dramatic when most of Down syndrome were living in institution. Because of increased propensity to infection and decrease in immune function, they die early. Now the picture is completely changed because most of children stay in family. They get better nutrition, and so the stress, the antigen stress, has decreased. However, a recent paper suggested that during the last influenza pandemic, there was a 3 times increase in mortality in subjects with Down syndrome, so it still is a big problem. The correlate with altered immune function is autoimmunity. There is unfortunately an increment of incidence of autoimmune disease in children and subjects with Down syndrome. For example, the increased incidence of diabetes, thyroid function, and is defective because there is an autoimmune response against the thyroid gland, inducing hypothyroidism. Also, it is important to remember that there is a high incidence, increasing incidence of celiac disease, which is an inflammatory disease of the gut. We can have also important consequence because the absorption of nutrients like vitamin can be compromised by the presence of this alteration. Of course, because of the decreased immune surveillance, like, for example, defective function of natural killer cell, there is an increased incidence of lymphoid and myeloid leukemia. Which are the weaknesses of these kind of pictures? We don't know if there is a primary immune deficiency in the syndrome or the immune defect we observe is a consequence of an accelerated aging, like a progressive deterioration of immune response. A recent paper published in Journal Immunology has suggested that probably Down syndrome is a good example, a typical example of non-monogenic immune deficiency. The overexpression of several genes in the chromosome 21 as a result of this immune defect starting early during the life. But since we don't live in a aseptical environment, we already battle every day with bacteria, viruses, and so on. The continuous stress that the environment puts on the immune system can also accelerate the deterioration of the system. Probably the real picture of Down syndrome immunity is a combination of these two factors. Non-monogenic immune deficiency and a contribution of accelerated aging of the immune system. Gaps to be filled, I just mentioned something simple but important. We don't know really how overexpression of gene in the chromosome 21 can influence the immune responses and cognitive function. There is a lot of exciting research going on, and so we need to clarify these aspects. Also, there is an important aspect that I'm studying in patients with Alzheimer's disease. Do acute and chronic or latent infection play a role in cognitive deterioration? Because it's a big issue, and we have exciting results from Alzheimer's disease, not in Down syndrome, but from Alzheimer's disease patients. And the other gaps to be filled is a big question. How can we improve the immune efficiency in children with Down syndrome? So let us just assess the first question. How many overexpression of chromosome 21 genes influence cognitive function? I like to make an example to keep this topic simple because it's very complex. Okay, we know, for instance, that there are regions very important on chromosome 21 that can replicate the trisomy of 21 in animal models or in other biological models. And for instance, we know that the Down syndrome critical region 1 is quite important because data are suggesting that genes in this small fragment of chromosome 21 can impair or decrease angiogenesis. And angiogenesis is a quite important function by a cognitive point of view. Why? Okay, let us see, for example, the neurovascular, the neural stem cell arrive during the development of nervous system, central nervous system, by vessels. So angiogenesis, continuous building up of angiogenesis in a developing brain is very important. And when the brain stops developing, the setting up of neurovascular unit, which is a complex picture composed of vessels, neurons, nursery cells, is very important for the efficient functioning of the nervous system. And then also the integrity of blood-brain barrier is controlled by angiogenesis. How much time do you have? 5 minutes, thank you. So there are biological and clinical consequences of impaired angiogenesis. We already mentioned about neurogenesis, nervous system development, and integrity of brain-blood barrier. But an impaired angiogenesis also disturb and alter inflammatory responses. So is an additional aspect of contributing to immune impairment. There are also positive effects. We can find this effect, clinical effect in the syndrome. For example, because of a decreased angiogenesis, we have a decreased atherosclerotic risk in subjects with Down syndrome, also a decreased growth of solid tumor. There is an increased incidence of leukemia and lymphoma, but a decreased incidence of solid tumors. Another question that I like just briefly assess is, do impaired immune response play a role in cognitive deterioration? Because there is a complex interplay between immune system and cognition and brain functioning. For example, we suggest with a couple of papers in Alzheimer's disease that individual genetic background, there are a lot of new genes coming from genome-wide association study. Most of these are part of ApoE4. Most of these genes are involved in immune response and defense against microorganisms. So the individual genetic background in Alzheimer's disease can affect the efficiency of immune responses against, for example, parasites like herpes that we are exposed to. Our life, 85–90% of old people, for example, are positive for one or plus virus of this family. And this herpes virus has the unpleasant capacity to infect the central nervous system and becoming latent. But when the immune system is weakened because we are under stress, we have bad diet, we are following a bad diet, or we have a concomitant other disease, the virus can reactivate. And through cycle of latency, reactivation can be, these vicious cycles can induce segmental, progressive macroencephalitis and kill neurons. And years after years can contribute to neurodegeneration in the brain of subject with Alzheimer's disease. Then, taking into account that subjects with Down syndrome have an impaired immune function, you can imagine that also chronic infection can play a role in the cognitive deterioration observed in the syndrome. How can we improve immune efficiency, especially in children with Down syndrome? There are several approaches. For example, a nutritional intervention protocol. We already listed several papers about this. There are exciting data coming from the Alzheimer field, can be applied also to the subject with Down syndrome. So I would suggest that it will be very exciting after, as a result of this meeting, that we suggest something to the scientific and social community and set up a multidisciplinary task with a title. I suggest a title just to give something exciting, like improve immune and cognitive performance in Down syndrome and prevention of the dementia in adults Down syndrome. Of course, we need funding and integration between basic and clinical research. This is a beautiful opportunity to have this kind of integration that is really lacking in the scientific community. So also, minding the gaps in Down syndrome, we need to talk with family a little more than we spend more time talking with family, because we need volunteers to participate, for example, to double-blind placebo trial for nutrition or other drugs we are able to suggest. And the end point is, let us try to increase immune efficiency, but probably that will be a good means to increase also, to decrease morbidity and prolong cognitive performance in adults with Down syndrome. And thank you for your attention.
Professor Licastro, maybe we are able to take one question or two.
The question was about a relationship about cognition and immune efficiency, okay?
Vaccination.
Oh, vaccination, excuse me. That's a big issue. I don't have the answer, but we cannot say no vaccination because they will expose children to very bad, very unpleasant disease, but we don't know if there is any relationship causal. But we can measure if the vaccination is effective or not. For example, if you give a vaccination against tetanus toxoid or something like that, you can measure the level of immunoglobulins in the blood, and so you can decide if that particular vaccination is effective or not.
That's what my question is. Is it quoted in the literature?
No, sorry.
Down syndrome respond as their peers to vaccination? That's the only question, because if you say that the immune system is defective, they should not respond to say vaccination.
There are data in the elderly non-Down syndrome subject. In the elderly about 70 years of age, if you give vaccination, the results are poor. So you have problem to increase the immune response and protect them against flu or whatever you like. Probably a similar situation is present in Down syndrome. We have to study this particular aspect.
Thank you.